All right, thank you, Moses. Okay, thank you, Moses, and thank you everyone for coming. Today is the second lecture about PET radiochemistry. So here is the outline for the two lectures. I'll briefly recap what we discussed in the last lecture and then tell you what we're gonna talk about today. If there are any questions from the last lecture, now is a good moment to ask. So in the last lecture, we talked about what radiochemists do and how radiochemistry is now an important aspect in PET tracer, in PET imaging and PET tracer development. We really need new tracers. Then we discussed what are the characteristics of good PET tracers. And these include having something that is metabolically stable, that is available that has high plasma availability, that it gets into the brain if you're trying to emit something in the brain, that it gets cleared fast, and how a lot of times you can correlate the physicochemical properties of your compound with the sort of the pharmacological property, and you can make changes to the compound in ways that affect those pharmacological properties in a sort of predictable manner. So if you have a compound that is not lipophilic, meaning it dissolves better in water than in an organic solvent, and you make it more lipophilic, that will typically improve your brain penetration, but that may also increase or slow down your clearance, for example. We talk about what are some of the common radionuclides for making PET tracers. There are many more. This lecture is mainly focused in small molecules, although today we'll see something about large molecules. We discussed about how some of these radionuclides are made. So gallium-68 is typically made in a generator, although it can also be made in a cyclotron. And then carbon-11, nitrogen-13, oxygen-15, F-18, those are pretty commonly made in a cyclotron. And then for labeling small molecules, we're really limited to carbon-11 and fluorine-18. We talked extensively about the chemistry of carbon-11 on Tuesday. We discussed the different reactions that can be done with carbon-11. Methylation is by far the most common, but there are others, and one has to think about carbon-11 as a very sort of powerful and versatile chemistry, because even though it only has a 20-minute half-life, we typically say that from beginning of synthesis until you start imaging, you have, say, 3 or 4 half-lives, so you have maybe 60 minutes, and in that time, you can do a lot of chemistry and purification. So the focus of today, we're going to spend the first half talking about F-18 chemistry. Then we're going to discuss some radiochemistry automation, and then I think for this audience that may be something interesting. There are also some opportunities for research and development in that area. Then we will talk about macromolecule labeling, and then end up with some sort of generic concepts that one has to keep in mind when talking about radio tracers. All right, so F-18 chemistry, there are 2 isotopes of fluorine. The F-18 is the natural isotope, it's non-radioactive, and F-18 is a radioisotope produced in a cyclotron, and it has a almost 2-hour half-life. So if you replace F19 for F18, that doesn't change the physicochemical properties of the molecule, other than now it becomes radioactive and it can be detected with a PET scanner. So when you add a fluorine to a molecule, what can happen? So a lot of times, and this is pretty recent, in the last 2 or 3 decades, the medicinal chemists are putting fluorine in many drugs. And the reason for that, it tends to improve drug potency, and because the carbon-fluorine bond is very strong, it also tends to improve metabolic stability. So those two are important benefits about fluorinated drugs. In addition, it can increase lipophilicity and log conformation of a molecule, which can result in sometimes beneficial properties. But the one thing to keep in mind is, say that you want to make a tracer and your original molecule does not have fluorine, then you have to find out or think about where you can put a fluorine to make it a tracer. But also you have to check that when you put the fluorine, the biological activity remains. In many cases, it will remain and maybe even be more potent and perhaps more selective. But in some cases, you put fluorine, and depending on where you put it, it's not active anymore. So those so that's why in radiochemistry, a lot of times people, the first generation tracers are labeled with carbon-11, and then the second generation, third generation are labeled with F18. So carbon-11, you can make the sort of a compound that you already know that is active, but have the limitation that the short half-life limitless applications. F18 has the advantage of a longer half-life, but you have to do a lot of work to make sure that that molecule is still suitable for your purposes. So in terms of the chemistry, so the way we think about this is we have a precursor, which is a molecule that does not have fluorine, it has something else. And then we want to put a fluorine in that molecule. So for the regular fluorination that a organic chemist may do, typically you use electrophilic or nucleophilic fluoride, most commonly electrophilic. Those are fluorine-containing reagents that are very reactive, so they work well with many different precursors. And then you use an excess of fluoride because your reactions are not decaying. You can do the reaction overnight. These electrophilic reagents are usually work in anhydrous solvents, meaning solvents that do not have any water. And then typically, because fluorine, the conditions to introduce fluorine are harsh, meaning high temperature, long reaction times, that other functional groups on the molecule may not tolerate. You typically introduce fluorine early in the molecule, and because the fluorine carbon bond is very stable, once it's introduced in the molecule, then it's robust. You don't have to worry. You can keep making modifications in the molecule and nothing happens to that fluorine. In comparison, when we want to make a F18 labeled compound, because of the short half-life, we have quite a few limitations. We have to put fluorine on the last step or almost the last step, which means that a lot of times we have to spend a lot of time building a very sort of fine-tuned precursor so that in 1 or 2 steps we can get to the desired product. We have to do the reactions quickly, and because we're using F18, the amount of fluoride is always limited. So the amount of precursor is much, much higher than the amount of fluoride in the reaction. So that can help the chemistry go forward, but that also means that you have to be able to separate your precursor, which may have chloride or iodide, from your product, which has fluoride. And then most often we use aqueous fluoride because that's what the cyclotron makes. So I'll talk about that now. So for F18 chemistry, we can do two types of reaction, electrophilic fluorination and nucleophilic fluorination. For electrophilic fluorination, that uses F2 gas. And this is, so if you remember from the previous slide, I talked that in cold chemistry, electrophilic reactions are favored because they're more reactive. So sometimes they use F2, or there are other fluorinating reagents that are electrophilic, but those cannot be produced in a cyclotron. So to produce electrophilic fluoride in a cyclotron, you have neon plus fluorine in your target, you bombard that with deuterons, kick out an alpha particle, and that generates F18, which recombined with the F19 to make some F18, F19 difluoride. And then this difluoride can be used for reactions. One thing to keep in mind is because your target gas already contains some fluoride, your molar activity is going to be very low. And the molar activity, we'll talk about this at the end, is how many, say you have 1,000 molecules in your source or in your dose, how many of those are radioactive versus non-radioactive. And so if all of them are radioactive, that's a very high molar activity. If only 1 out of 1,000 is radioactive, that's a low molar activity. And you can imagine that if you have a large excess of non-labeled molecule versus labeled molecule, those are still going to bind to the target, and they may block the specific binding. So the more common reaction, or the more common source of fluoride is nucleophilic fluoride. You make that out of O18 water by the PN reaction. And then, so on your target after bombardment, you have a few F18 anions in water. And then because the F18 is negatively charged and the water is neutral, you can then trap the F18 on a cartridge that has a positive charge, on a solid support that has a positive charge. And then you can elute it in an organic solvent. So usually you still have a little bit of water left in your fluoride, which is going to sort of limit the reactivity, but that's the best we can do, basically. And the advantage of this method is you can get high molar activity. All right, so one example of electrophilic chemistry, this is F-DOPA. F-DOPA is an analog of DOPA. It interacts with dopaminergic neurons, and it can be used in imaging neurodegenerative disorders like Parkinson. And then, so here is the example of a synthesis. It's a simple synthesis. You have the teen precursor. You have the teen precursor, which reacts with F2. You get direct fluorination on that position, and then you have to deprotect all the other functional groups to generate F-DOPA. The reaction is highly efficient and is fast. The molar activity is low, but in the case of F-DOPA, the molar activity doesn't matter so much because you're not looking at reversible binding, you're looking at accumulation in those neurons. So and then if we go to nucleophilic fluorination, I already alluded to this, but basically you have F18 minus and O18 water from the target that get loaded into a cartridge. The cartridge has this sort of trimethylamine motifs that are positively charged. So the water will go right through, the F18 gets trapped, then you can elute this with a counter ion. So typically carbonate or bicarbonate. And then you also use this K222. This is a krypton that chelates the potassium and makes the fluoride more soluble. So, and then having little water in your final solution is important in order to get good reactivity. All right, what about how does the nucleophilic fluorination work? So the first type of reaction is nucleophilic aliphatic substitution. You may have studied this in basic organic chemistry. So you have a aliphatic carbon with the 4 substituents, one of them being a leaving group. Fluoride will attack from the opposite side to the leaving group, and then the leaving group gets kicked out and you get inversion of the stereocenter. So that's the traditional SN2 mechanism. It works with aliphatic carbons. And then as leaving groups, typically we use triflate, mesylate, tosylate, nosylate. Those are the common leaving groups. And then, so here are 2 examples of aliphatic, nucleophilic aliphatic substitution. So this is FDG, by far the most common tracer. So in this case, we have a precursor that is basically on the 2 hydroxy position, we have a mesylate, and then all the other hydroxy groups are protected as acetals. And then you add the fluoride in cryptofix, 85-degree acetonitrile, and in a matter of minutes, you get fluorine incorporation here, and then you hydrolyze the other positions. Here is another example of nucleophilic aliphatic substitution. In this case, we're making FLT. FLT is fluorothymidine, is a sort of precursor of DNA, and it is a marker of proliferation. So FDG is a marker of metabolism, and you can detect highly metabolically tissues or cells, including tumors, and then FLT is a marker of proliferation. So you can tell things that are sort of dividing quickly. So in this case, how the chemistry works is, again, you have these nosylate leaving group where the fluorination is going to occur, and then you have the other hydroxy and amine groups which are protected with bulk. So fluorination occurs first, and then you get the protection to make FLT. All right, so yeah, the second sort of reaction with nucleophilic fluoride is nucleophilic aromatic substitution. This is when you're trying to make an aromatic fluoride, so meaning fluoride in a benzyl or phenyl or heteroaryl group. So in this case, this works best if you have an electron withdrawing group on your aromatic ring, because F18 is not a very good nucleophile, and if your aromatic ring is electron rich, meaning it has sort of high negative density, the fluoride is not going to attack yet, because there are opposite interactions. So if you have an electron withdrawing group, which is sort of pulling electron density from the ring, and you have a partial positive charge in here, you have more chances to get the reaction. And so this is the typical mechanism. You get this sort of intermediate where you have both leaving group and fluoride attached to the molecule, and then the fluoride stays, the leaving group leaves. So the common leaving groups are these trimethylamine, positively charged, this dimethyl sulfur, nitro, iodine, bromide, chloride, methoxy, tosylate, triflate, etc. So I already said it works better with electron withdrawing substituents. These include nitro, cyanide or nitrile, trifluoromethyl, carbonyl groups, etc. And this can be done either metal-free or with some metal catalysis. All right, so here is an example, and this is, you'll remember this from a couple of the slides ago, this is F-DOPA. But if you remember from the previous slide, this was a two-step reaction. You have good reactivity, fluorination is step 1, the protection is step 2. To make F-DOPA via with nucleophilic fluoride, it takes many more steps. And the reason for that is, in this case, the aromatic ring does not have a electron. So in the final product, which is this one, the aromatic ring does not have electron withdrawing groups to make this sensitive enough for nucleophilic fluorination. So what you have to do is to start with this sort of precursor where you can do fluorination, and then you have to do multiple steps to couple this with the other half of the molecule to finally generate, and then the protection to finally generate F-DOPA. So many steps. This is a big accomplishment that after so many steps, you get a yield around 20%. All right, here is an example of aromatic fluorination using iodonium salts. These are not so easy to make, but if you can make a precursor that contains this sort of motif, diaryl iodonium salt, this is basically a hypervalent iodine that has a positive charge. These undergo fluorination usually pretty efficiently and under mild conditions. And this is another version of that same chemistry or similar chemistry. So another example, in this case, using tin compounds. Here the copper is required for the reaction to take place, and these reactions have high specific activity as most of the nucleophilic fluorination, and it has good substrate scope. Here's an example from my own work, and this is, we wanted to make 3-fluoroaminopyridine. This is a tracer that we're investigating for multiple sclerosis and other demyelinating diseases. It binds to potassium channels in the brain, and then to make this compound, the fluorine is on a pyridine that is very electron rich. In addition, we're trying to put fluorine in the meta position. So this is ortho-meta-para, which is the least favorable position for fluorination. So the way we got around that problem was, rather than doing fluorination on the pyridine directly, doing fluorination on the pyridine and oxide. So pyridines, they can be oxidized to this type of motif. This an oxide, sometimes drawn as a double bond or as a single bond with a positive and a negative charge. That sort of changes the electronics of the molecule, making it much more sensitive to nucleophilic reaction in the meta position. So here we can do fluorination under very mild conditions on the meta position and then get the desired compound. The yield of this reaction was not very high, because if you heat up the reaction to try to increase the yield, now you get competing reaction in the nitro, which can also act as a leaving group. So, and then this is, yes, a sort of a test reaction showing that the presence of the nitro group is critical for the reaction to occur. If you just have the bromopyridine and you try to do fluorination, you get nothing. But if you have the nitro, then you can get efficient fluorination. Another example of FAT in chemistry, which is not very common, but I thought it was kind of cool, is using this enzymatic fluorination enzyme called fluorinase. So fluoride is not very common in nature, and there are many natural products that contain fluoride, and there are many enzymes that work with fluoride. But this is, I don't know, some unique enzyme from bacteria that can catalyze the incorporation of fluoride. So the interesting thing about this reaction is that it actually works in water, like all the enzymes, but fluorination reactions, they almost never work in water. So that sort of tells you how powerful this enzyme is. Well, let's talk a little bit about radiochemistry automation. So when we do radiochemistry, typically we would, if we want to make, say, a dose for a human or a large animal study, we need to have, say, 20 to 50 millicuries at the end of the synthesis. If a synthesis takes 2 hours, then it has, say, 50% yield, then you need to have, instead of 50 millicurie, you're going to need to start with 400 or more millicurie to start. That is a very high dose to handle by hand. So we use this type of equipment. This is basically a chemical fume hood that is surrounded by lead shielding. So in this case, there's a door. Inside you have your radiochemistry equipment, and then the reaction is controlled using a computer, basically. So how does the synthesizer look like? This is an example. Since I didn't show this during the carbon-11 lecture, I will show it here. But this is a picture of how the synthesizer looks like for carbon-11. You have sorry about that. You have the reactor in here, and then the final product comes out of one of the little tubes here, and then these are valves that control the flow between the different parts of the reactor. And then your computer software, it basically looks like this. So you have the input from the cytosol, which will come here, carbon-11 CO2, and then it will go into an oven that traps the CO2, and at the same time you will flow hydrogen from a hydrogen tank. You react the CO2 with hydrogen at 400 degrees. You have some metal catalyst, usually nickel, in the oven that will make CH4. And then the CH4, you further send it here. Here there's a cooling trap, meaning you have liquid nitrogen that is cooling a sort of a metal coil. And the trap, you can control the temperature using software, basically telling how much liquid nitrogen to use. So I think this is typically during a synthesis like -70 or -80 degrees. So you'll send the methane into the trap. If there's any remaining CO2, it will get trapped in the sodium hydroxide scavenger, and then the methane will be condensed in the liquid nitrogen trap. Once you've collected all the methane, and you have basically radioactive detectors throughout the different pieces, so here is one, here is another, so you can monitor how the radioactivity is being collected. Then what you do is you increase the temperature of the trap so that the methane is released. So it's released into this loop, and then here you have an oven where you volatilize iodine, and then here you have another oven where the iodine will react with the methane. And because this reaction is not very efficient, it doesn't happen on a single pass. So you have to recirculate over and over for a few minutes the methane plus iodine mixture to generate methyl iodide. And then you have here a trap that collects the methyl iodide. and you have another detector. So once you see that your methyl iodide has been produced, then the circulation stops and it comes out of one of these lines, basically. And that will go into the vial where you're going to do the methylation reaction. Here is another example of an automated system for, in this case, trifluoromethylation. This is a homemade system that we set up here at MGH to make trifluoromethyl labeled with carbon 11. And in this case, we get methane from the previous system. So you can see here that you get methane formation here. And then typically you will go all the way to methyl iodide, but you can stop the reaction here and then send the methane out through the line. So you take methane, you have valves that allow you to control the flow where it goes, and then we have detectors. So first we pass the methane through a phosphorus pentoxide trap. This will trap any moisture that is present in the gas because moisture will kill the reaction. And then it goes into a column that contains cobalt trifluoride. The methane reacts with the cobalt trifluoride to generate trifluoromethyl so that you can collect in a cold trap. And we have detectors so we can monitor the radioactivity going through. So as we see the activity passing from detector 1, we can switch the valve to open, then we see that it passes through detector 2, then we see how it accumulates in detector 3. And then at this point, it means we stopped the synthesis and we took out the activity to do reactions. And this produces trifluoromethyl in very high purity. And then here is a picture of how the system works. So here is the Zika trap shown here. Here is the column with the cobalt trifluoride where the reaction happens. Here is a trap. This is the coil trap to trap any hydrogen fluoride that is formed. And then here is the reaction vial where it gets collected. And then we take that vial for additional chemistry. And all of this is inside of one of those hot cells so that the operator is just outside toggling the valves. Here is an example of a box for F18. This is how it looks. So F18 chemistry tends to be more complex. So in this box, we have many different vessels containing the different reagents. And we have 2 reactors and a large vial here for formulating the final dose. So again, the F18 from the target will come into the F18 vial. From there, it gets loaded into the QMA cartridge that it will separate the O18 water from the fluoride. Then you add your cryptofix in acetonitrile to elute the fluoride into the reactor. And then you add the precursor to the reactor. You can control the temperature and the pressure on the reactor. You do the reaction. After you're done, if you have multiple steps, you may add other reagents. And then at the end, once you're done, this has an HPLC attached to it. So you can send it into your HPLC column and you can monitor you can do a chromatography where you can monitor the different sort of reaction products coming out, and you have both UV and radioactive detection on the HPLC. So when you see your peak that it comes out, you just toggle the valve and you switch from your waste into your formulation vial. And then the formulation vial, you may, depending on what your HPLC solvent is, you may use directly for injection, or you may load it into another cartridge and then elute with something like ethanol that is biocompatible. All right, comment on macromolecule labeling. So up until now, we discussed a small molecule labeling. Small molecules have some advantages, like for example, if you want to do imaging in the brain, it's very hard to get a large molecule into the brain. If you want to image, I don't know, enzymes or things like that, it's easier with a small molecule than with a macromolecule. But macromolecules have advantages as you can have sort of peptides and proteins that they sort of mimic the endogenous protein or the endogenous peptide that binds to the natural ligand. So for membrane receptors and hormones and things like that, macromolecules may work better. And before I go into the macromolecules, any questions on the F18 chemistry or small molecule labeling? All right, so we'll continue with macromolecules. So when we talk about macromolecules, as I said, we typically think of peptides and proteins. A small molecule, it may be, I don't know, around 500 Daltons. A peptide may be a few thousand. A protein, so a few thousand meaning like 2,000 or 3,000. A protein may be tens of thousands, like, I don't know, 50,000 to 250,000 Daltons, typically. So because these molecules are large, you can think of them as a sort of a transatlantic vessel going through your bloodstream, as compared to a like a fighter jet, which would be a small molecule. So they move very slowly, and that has two important repercussions. One is they're going to be in circulation for a long time. So you inject them in circulation, they will eventually bind to the target and clear from circulation. But that process, if you're talking about an antibody, it may take several days. So you need a radionuclide that whose half-life matches that of the molecule. The other advantage of using, or one difference about using macromolecules is that since the molecule is already quite big, typically if you add a chelator, which is something like this, and a radiometal, so you're adding, I don't know, this may be 400 or 500 Daltons, maybe less, maybe 200, but if you add something like that to a small molecule, that's going to change a lot the pharmacology. Whereas if you add that to a protein or a peptide, then it has minimal effects on the pharmacology. So, and then here is an example. This is a cyclic peptide, and here's an example of gallium-68 label. And I don't know if this is DOTA or I don't remember, but I think it's one of those peptides to image some sort of hormonal receptor. All right, so one method on the previous example, we saw a way to label macromolecules by having a chelator and a metal. Another way to label macromolecules is using click chemistry, and then this uses what is typically called a click reaction, which means it's a bioorthogonal reaction that occurs very quickly. So in this case, you may have a macromolecule shown here as a green square with an A side, and then which react with an alkyne to make a tetrazole. So if you have a radioisotope on the alkyne, that can react with this and generate your label molecule. So this allows to make sort of modular changes. You can use the same molecule for many different large molecules, etc. So one advantage of this type of chemistry, and there are different versions, which I think I have on the next slide, are that this can happen inside of the body. So you can inject the macromolecule that does not have the radio label, and then you can inject the label, sort of the other half that is radio label, and it can be F18 because you inject this, then you can wait a day or two for it to bind to the target and clear from circulation, then you inject the label motif, and the reaction happens, and you can see where the macromolecule was located. Here are some other examples of molecules that people have made for click chemistry. For click chemistry, depending on the motif that you have, it will be more or less reactive. It will give you some sort of distance between the reactive side and the label, which may be beneficial as it will not interfere with the biological activity. So here are a few examples of alkynes and acids that have been used in F18 click chemistry. All right, another example of macromolecule labeling is using this type of bioconjugation. So in this example, what they are taking advantage of is of the very high binding affinity of fluoride towards silicon. So you can make the F18 silicon reagent and then react that selectively with a macromolecule that has a reactive group like a malimidide or something like that. Here's an example that I believe Moses was involved in, which is using heat-induced radiolabeling to label nanoparticles. So in this case, they were interested in making what is called ferroheme, is a nanoparticle that is used as a MRI contrast agent. So it has an iron core. And then what they develop is a clever strategy for radiolabeling with zirconium-89. And they call it heat-induced radiolabeling. So you have the nanoparticle, you put it under high temperature with a few atoms of zirconium-89, and those will get incorporated into the core of the nanoparticle, making now zirconium ferroheme, which is radioactive. Furthermore, they can modify these molecules to add additional functionality. So either by doing click chemistry, like what we just discussed, or directly putting a label on it. It doesn't need to be through click chemistry. And the advantage of this, it allows you now to target the radiolabeled nanoparticle to specific cells, for example. So if you just give the zirconium-89 ferroheme to animals, then any sort of phagocytic cell will take it. But if you have a specific receptor on the surface of the nanoparticle, it can be selective for some cells. All right, here is an example of cellular labeling that I think is quite interesting, and it sort of tries to accomplish something similar to the previous example. So the previous example with the zirconium-89 ferroheme is used for cellular labeling. So one thing you can do is take cells outside of the body, label them outside of the body, and then reintroduce them in the body. But one thing that some people would say is, well, as cells divide, the amount of activity also divides. So there are only so many divisions where you can still track the radiolabeled particle. So what these researchers did is sort of a genetic approach where you can take cells and then engineer the cells to express this sodium iodide symporter. And then how the sodium iodide symporter is an endogenous molecule, but is only expressed in the thyroid. So is this protein is meant for sort of taking iodide from the bloodstream into the thyroid to make the thyroid hormone. But if you take that protein and then you add through some genetic manipulation, you add it to the cells of interest, now you have sort of these immune cells that you may be interested to use for cancer treatment, that now they have a handle for you to image. And the way these are imaged is that you reintroduce the cells in the body. Now they're expressing this protein. So if you then administer to the subject F18 BF4 or F18 SO3F, these are large anions that they sort of mimic iodide ion, and then they're going to be taken up by the sodium iodide importer, and those cells are going to light up. And I don't know how commonly used this is, but it's a clever technique of genetic engineering for imaging cells that have been transplanted into the body. All right, so a few final slides on some important concepts about PET radiotracers. One is molar activity. Molar activity, as I mentioned before, refers to the ratio of non-radioactive molecules per radioactive molecule. And so typically, this was an in-person class, we would calculate the theoretical maximum molar activity for F18. But I'll let you do this on your own. But it's basically 1700 curies per micromole. When we make F18 tracers in the lab, the typical molar activity for F18 is 1 to 5 curies per micromole. So that means that on average, 1 out of 1,000 molecules are radiolabeled. And by the way, if you want to calculate the theoretical molar activity, you can integrate these integrations per second using the half-life of the fluoride, and then see how many atoms of fluoride you have in a mole of fluoride using Avogadro's number, and you should be able to get to this number. Is an interesting exercise. And so going back to here, F18 tracers, they have a molar activity between 1 and 5 curies per micromole. So most of what we inject is not radiolabeled, but a molar activity of 1 to 5, we call it, or anything greater than 1 curie per micromole, we call it as no carrier added synthesis. And that's typically enough to image even very low abundant receptors. If that drops by 10 or 100-fold because you have a synthesis that is carrier added, meaning you have fluoride on other fluoride, you're doing a fluoride exchange, for example, then you're going to have difficulty detecting low abundant receptors, because now the non-radiolabeled molecules are really going to block most of the receptors, and then the labeled compound, the labeled molecules, the fraction of labeled compound, they're almost not going to have enough available sites to bind to. For carbon-11, the theoretical molar activity is higher, and you can think about why is it higher? Can you correlate that with the sort of half-life of these two molecules? But eventually we end up with a relatively similar number of labeled to unlabeled compounds. So this is an important point that you'll hear a lot of radiochemists talk about, saying like, oh, this synthesis is not good because it has low molar activity, or this synthesis is great because it has very high molar activity. The molar activity is not critical for all the tracers. FDG, it doesn't really matter. F-DOPA, it doesn't matter either. But it is important for sort of the traditional receptor reversible binding tracers. And you can think about why that may be the case. And then another thing that is important to consider when sort of making new radio tracers or planning to use radio tracers is how they are metabolized. And I think next week you have the lectures on pharmacokinetic modeling, and Mark Normandin will discuss why the metabolism is important. But basically, the most molecules that you give to a subject, they get metabolized, right? The body is very good at taking a molecule, and if it's something they can use for convert it to energy or nutrient or something like that, they will break it down and use it. If it's something they cannot use, they'll break it down and excrete it. So during that breakdown process, if your molecule is radioactive, what it means is that your radioactivity does not disappear, yes, because the molecule is broken down. What it means is you have the radionuclide on a different molecule, which is typically an oxidized version of the molecule. And most often than not, that radiometabolite will not bind to the target. So it's just going to contribute noise to your system. And even if it binds to the target, it's still problematic because a lot of times you don't know how much of the molecule has converted. And this metabolic conversion is also time dependent and is very subject dependent. So there might be people that metabolize molecules very quickly, and there might be others that do not. And so the rate of metabolism changes from person to person. So tracers that get metabolized, they tend to be problematic because you need to measure how much of the tracer remains in blood at any given time. In some people would say that in some cases metabolism is useful because it helps you eliminate the drug faster. And so in the cases where, say, your drug goes in, binds to the receptor partially, whatever is not bound, it gets metabolized and it gets excreted into the urine, and then you take an image later, that could help you get a higher signal to noise. You still have to make sure that that's the case. You need to measure the amount of parent compound in plasma and make sure that it's giving you a stable and robust signal across many subjects. And then we talked in this lecture about nucleophilic and aromatic fluorination. Nucleophilic fluorination, meaning fluoride in alkanes, they tend to be less stable in vivo. It's easier to break those bonds, and that will lead to defluorination. Whereas aromatic fluorides, they tend to be more stable, at least to defluorination. Those molecules may still not be stable, but they're probably not going to undergo defluorination. And when a tracer undergoes defluorination, then you have free fluoride floating around, which accumulates in bone and teeth. Here is an example of a molecule called whey that undergoes metabolism in vivo. And you can see that depending on where you have the label, you're going to get different imaging results. So on the example on the left, you have the O-methyl carbon 11 whey, meaning the carbon 11 methyl is on the methoxy group on the phenyl ring on the left. So when there is metabolism, that molecule breaks in two. So basically the amide bond here will hydrolyze and will generate this carboxylic acid, with this carboxylic acid. So now you have this new radiolabeled molecule which gets into the brain and it binds to the target, but it probably doesn't bind with the same affinity, and may give you some confounding results. Now, if you have the carbon 11 label in this position, the molecule is still going to undergo the same type of metabolism. So it's going to, the amide bond here is going to break, but now your carbon 11 label is in this other piece, which is not brain penetrant and not pharmacologically active. So if your label is here, all you're going to see on the images is this one. Well, so at the initial state, once this has cleared from circulation, this is all you're going to see on the images, whereas in this case, you're going to see both. And then here, the researchers developed a fluorinated version, and they decided to put the fluoride in this part of the molecule, hoping that they would get the same as here. So it doesn't matter if you get cleavage here, it's not going to affect your images. But what happened here is that you get defluorination of the parent molecule, and then your images are also going to be problematic. So to summarize, one thing that radiochemists say, that if it is easy to make, it's probably not a good tracer. I think there's some truth to that, and the reason is chemical reactions that are easy to undergo are usually to make bonds that are not very strong. Those are the ones that are easy to break by the body. So if you want to make a tracer that is very robust, it's going to be harder to synthesize. And then here are some resources if you want more information. My email is also there. I'm happy to discuss more if there's interest. And I think this is the end of my lecture. It is a little bit shorter than it usually is, because typically we do some exercises in the classroom, but since we're doing it online, then it's a little shorter. But that means we have a few minutes to take questions, and once we're done with questions, we'll yes, sort of make the lecture shorter today. All right, thank you for your attention. Any questions on what we saw today?